Hi everyone, and welcome to our first installment of the Gene Delivery Seminar Series for 2026. Yay! I'm very excited to introduce you to our seminar speaker today, who's Dr. Cristina Martinez Gonzalez. Cristina is a neuroscientist and research fellow at the University of Edinburgh, whose work has advanced our understanding of autism spectrum disorders, epilepsy, and also spinal cord injury. She combines optical tissue clearing with light sheet microscopy to map neuronal architecture and circuit alterations in rodent models, enabling unbiased 3D analysis of intact brains and other organs. Cristina previously held postdoctoral roles at both the University of Edinburgh with Professors Matthew Nolan and Katerina Becker, and at EPFL in Professor Gregory Courtine's group. She earned a PhD in neurosciences from the University of Oxford, as well as a master's and bachelor's degree from UNAM, Mexico. She's part of the light sheet microscopy and 3D analysis team, which supports researchers across the University of Edinburgh and the Simons Initiative for the Developing Brain, with tissue clearing, light sheet imaging, and 3D image analysis. Today, Dr. Cristina will present tissue clearing protocols for diverse organisms, sample types, and applications, sharing core principles and practical guidance for choosing the right clearing approach for different samples and to meet your research goals. So I would also like to remind everyone to put questions in the Q&A box, which Cristina can answer at the end of her talk. So yeah, thank you everyone for attending. And yeah, Cristina, please take it away.
Thank you very much. And thank you very much for the invitation from Vector Builder to me to give this talk. As Lindsay, I'm Dr. Martinez Gonzalez. I work at the University of Edinburgh, and I'm a research fellow there. And I established and now lead the light sheet microscopy facility. So I just want to say a very quick overview of what is going to be my talk. First, I'm going to talk about why tissue clearing, and then I'm going to move towards types of clearing methods and what are their advantages and disadvantages, particularly for different types of models, different organisms, different structures, and what are the tissue processing steps in general. To start, why tissue clearing? It's basically because normally any biological sample is not transparent. It's always embedded as lipids, tissue, blood, protein, and it is not possible to analyze in depth using a microscope. So any biological samples normally will have all these components, and these components, particularly lipids, affect the way the light passes through it. So to see inside the tissues without actually sectioning, we need to clear it. Because before light sheet microscopy and tissue clearing, the methods for analyzing brain tissue were very labor intensive and provided very limited information. You needed to slice the tissue in coronal sections, horizontal sections, sagittal sections, and you will have a huge amount of planes. And at the end, no matter what, the way you cut the tissue, you will end up with limited information. You will never get the entire picture. And we think why obtaining this three-dimensional information is important. And for that, I'm going to show my case with this piece of art called the square in the circle. If you look at the structure in one plane, you will never get the entire information. If you look at the information in 3D, in all the angles, you will realize it's not a square, it's not a circle, it's not like a set structure. Same with the brain. All our structures in the brain, we are used to see them in coronal planes, in sagittal brains, but you will always miss information unless you actually clear the tissue and look at it in its entirety. So this will allow us to see, tissue clearing will allow us to see intact structures, intact organisms as well. And tissue clearing can improve your research in many ways because it will allow you to image deep into the fixed tissue by rendering transparent. So basically, any sample that is larger than 50 micrometers thickness will benefit from tissue clearing. And the question of the hour, how do we decide which tissue clearing method is really optimal for your research? It's complicated. It's not like a one map that you can follow. Basically, there's quite a lot of clearing methods right now, new techniques, and each one of these will have a trade-off between how fast the protocol happens, how expensive it is, how complex, basically how much experience you will need in tissue clearing, if you have fluorescence or not, if you like to do an immunolabeling or not, and etc. So the question, your biological question, your scientific question is the most important part to determine which kind of microscopy and which kind of tissue clearing you should be using. And my entire talk, I'm going to mention the large families of type of tissue clearing, and then I will tell more or less which are the most suitable for certain species, more suitable for different type of labelings, etc. But again, this is never going to be like a one fit everyone. So moving towards my tissue clearing methods and each advantages and disadvantages. Last time we looked, about a year ago, I checked literature in a review. There are at least 100 optical clearing methods out there in the literature. This is a review by Ueda et al. And divides the optical clearing methods in three large families: hydrophobic methods, hydrophilic methods, and tissue transformative or hydrogel embedding methods. And I'm going to speak to each one of those three families briefly to explain what are they and what are the steps on it and what are more useful. The first one, hydrophobic tissue clearings, or much well known as solvent-based clearing, organic solvent-based clearing, includes started with the 3DISCO clearing methods, and they are the most common ones out there. Any one disco of the disco family they are the most popular methods at the moment, including our own method I will very briefly talk at the end, called RatDISCO, which is a tissue following the hype of the disco methods. It's called UDisco, VDisco, etc. So we call it RatDISCO because it's basically defined for large rodent brains. And I will talk a bit about that method at the end. And basically, the principles of hydrophobic or solvent-based includes dehydration steps of the tissue. We need to remove all the water to start with. Then we can do antibody labeling, which is optional. We can label any or not just antibody labeling. We can use dyes or to label different structures. Then we need to remove all the lipids because the lipids is what actually make the light to scatter through the tissue. This step is called delipidation. And finally, we do something called refractive index matching. I'm going to briefly show at the end what exactly do I mean by that. But this is a step which makes the tissue, renders the tissue optically transparent. And the main purpose of tissue clearing is that when the tissue is transparent, we can use not just the traditional confocal microscopy, which is limited to very, very small working distances. For this kind of tissue preparations, ideally, we recommend light sheet microscopy because it can image through millimeters of tissue and without very little photo bleaching, and which very fast and with very large fields of view. But this talk is not about microscopy, it's just about the clearing methods. But if you ever clear your tissue, I would recommend to use light sheet microscopy rather than the confocals. So, how suitable is hydrophobic tissue clearing? Well, it has huge advantage. I'm showing one example in here of a half brain of a mouse that we immunolabel against FOXP2, a transcription factor. We're using our RatDISCO protocol. And the huge advantage is very fast. Clearing is fast. Antibody labeling may take a bit of time, but the clearing is a process of a day or two maximum. It's very economical, it's very cheap. The solvents used for this method can be really, really cheap. The samples are, when you finish with your clearing, the samples are solid, like they look like glass, and they will last forever. In our facility, we had image samples that we produced 8 years ago because someone came back with a new question to image in a different part of a whole brain, and we re-imaged the tissue, and the tissue is intact. So you can image and re-image, especially for people who do large behavioral cohorts. You can reuse the samples. Imagine you get your data, you analyze it, you submit your paper, and then 3 years later you need to go back for reviewers' comments. You can easily re-image your tissue, which is for us is a huge advantage. Unfortunately, the tissue, because it's solid like glass, it's a bit fragile. So if you accidentally drop it, well, that is a huge problem. Solvents, the solvents used can be a bit hazardous. So equipment needed like fume hoods and um basically, fume hoods are essential. It cannot be done just on the bench, but most labs will have fume hoods. And this kind of methodology often will destroy the endogenous fluorescence. So if you have a transgenic mice, if you clear it, the fluorescence will die. So basically, you need to use an antibody step to bring back that fluorescence labeling, and that fluorescence will last forever, which normally is not a problem. But some people might want their antibody. They would not want to use antibodies, so perhaps the antibody doesn't exist. In that case, these methods perhaps are not ideal. And finally, tissue shrinkage. You can have a little bit of shrinkage or a lot of shrinkage, and for some people might be a disadvantage. But for me, for example, that I work with rats, the rat tissue is huge. The brain of rats the size of my thumb is massive. So finding a microscope with enough working distance to put it on there is not that easy. So shrinking the tissue for me is quite advantage because a smaller tissue means a smaller area to image, small data. At the end, I don't end up with terabytes, I end up with gigabytes, and for me it's an advantage. But if you already start in an organoid, perhaps it's not ideal that it shrinks even more. Um, so for this, I would say this method is suitable for large biological samples, whole embryos, whole mouse, large brains, hearts, etc., kidneys, the whole structures. Um, The second very large family of tissue clearing methods are the hydrophilic tissue clearings, which basically are water-based. There are several out there. One of the most popular are cubics. They use either water-based or hyperhydrating solutions such as sucrose or urea to expand or to basically increase the tissue. Um, uh, yeah, to increase the transparency of the tissue. And this is one of the most popular and most used methods right now called cubic, in which they can clear the whole organ, the whole mouse in this case, for example, and they can use heart, lungs, etc. The general principle for this particular method, the cubic one, is washing the tissue, of course, in PBS. They remove the pigments by trying to remove the pigmentation of the skin, the hemoglobin, etc. Once they have removed the color, they do then remove the lipids. After this step of the delipidation, they can do antibody labeling again, and finally refractive index matching, which is very similar. All those principles of clearing are similar across methods. The difference is the solutions you use. In this case, you can use sucrose, urea, etc. In solvent base, you can use solvents like tetrahydrofuran, etc. And the advantages of this methodology is they are water-based methods, so they are not as hazardous as using solvents. So some people might feel much easier using this. It reduces autofluorescence in the images, mostly because of those the step I show when they remove the blood or any color, any pigments on the tissue. They are compatible with antibodies, but they are not necessary. So because they can preserve the endogenous fluorescence, they were very suitable for tissue which it has endogenous fluorescence, transgenics, etc., or diet label, viral injected animals that will be as well useful for this without the need of extra antibody labeling. However, some of the steps can diminish some of the fluorescence because of this is a water-based approach. It's definitely not compatible for lipid stainings because so if your molecule of interest is attached to the lipid at the membranes, it's probably not ideal for that. And the solutions at the end are very viscous. The sucrose and the urea are really, really viscous. It's difficult to handle. So that can put some people off for that. And the same as solvents clearing, these methods can shrink the tissue. For us would be an advantage, for some people might not. But the basically depends on the sample you start with. And this will be very useful for, again, lipid-rich tissues such as brain, such as tumor, small samples, but it would definitely not be suitable for anything that you want to label the lipids for. And the third large group of tissue clearing methods out there is the hydrogel embedding one. The most famous one out there is Clarity. And what they do in this case is they embed the tissue into a hydrogel. Basically, they create a scaffolding inside the protein. And then, like in here, you can see they infuse the tissue with a monomer. It can be acrylamide with bisacrylamide, or there are many other hydrogels out there. And once the tissue is embedded in this scaffolding, they use soap, basically sodium dodictyl sulfate micelles, to remove all the lipids. So it's basically the same as other methods. Removing the lipids is the most important part. You remove the lipids with soap, little by the micelles made of the soap. There are ways to make it active, and the way normally in passive clearing. And this is the general principle. You need to start with either a transgenic animal or use an antibody labeling. Then immerse your tissue into this hydrogel polymerization. And then it's a bit sometimes complicated how you make the mesh within the tissue, because if you tighten the mesh very much, it will take a long time to clear. If you make it too loose, your tissue will disintegrate. So this method has a lot of trade-offs out there. If you make the passive tissue clearing, which is literally just washing the tissue with soap constantly, the tissue will keep quite a lot of its integrity. However, it will take months to clear. A mouse brain can take a month or more. A rat brain will take months and months and months to clear. And there are ways to increase that with electricity. Basically, they base in this concept of making a Western blot gel, because the tissue is embedded in acrylamide. If you pass electrical current, you can remove not just the lipids with the soap, but you remove some of the proteins and speed up the project. Unfortunately, if you remove your proteins with electricity and you try to see something in there, you will lose your proteins basically. So it's basically making your brain a huge Western blot gel. Um, it has advantages, of course. For example, it's being water-based is helpful for many things. However, it's toxic, because acrylamide and misacrylamide, they are toxic. Hydrogels are quite toxic in general. Tissue will expand, but it can be reduced. For example, this is an example of a half a brain of a transgenic mouse that I took it to the Carl Leiser lab during his Clarity training courses, and it was imaged in the cone. Because the tissue expands, you can have huge resolution, and that improves. If, for example, here you can see neurons in the, I hope you can see my laser or my, here you can see neurons in the medial entorhinal cortex and the perirhinal cortex, and they sending projections all the way around the hippocampus, up and back, innervating part of the thalamus. So in 3D, you can see how nicely they look. And this example I like to show to mention, I was working with these transgenic mice before clearing for about a year. I cut it in every possible angle, coronal, sagittal, horizontal, And no matter which way I cut, I would be able to visualize those axons because they go vertical, they turn around, up, backwards, and innervate the thalamus. So this is a great example to show why clearing works. And after a year of shopping tissue, got nowhere, I took 1 hour to take this image, and I got exactly where these connections were going. So it's really, clearing is great for this. And water-based, especially these hydrogel embedding methods, like this image I got, it was beautiful. However, tissue is difficult to handle because you're talking about a tissue which is a gel. So basically, it can disintegrate like a gum. It starts with a gummy bear structure. Until time passes, it will flatten out like a collapsed jello or jelly. It will completely disintegrate. So once your tissue is ready to image, you have a very limited time frame to image, and then the tissue will be lost. It has been historically known to be very badly for antibody penetration and antibody labeling, particularly in large samples. So if your sample is transgenic and fluoresce on its own, is great. If not, it's definitely not suitable. Again, it's very time consuming. You can lose a lot of protein with this approach, and the tissue won't shrink. The opposite, the tissue will expand in size, which if your sample is small, like an organoid or a mouse brain, is okay. But if I try to expand a rat brain, there is no microscope out there that will easily image my entire sample. So, and of course, large volumes, much, much more data, which is something to consider. So we recommend this kind of clearing approach for small samples, particularly things like zebrafish, spinal cord. This is kind of suitable. Any larger than that, mouse brain is a stretch. And on that note, I know this is not tissue clearing, but based on the clarity, the very, very similar approach is expansion microscopy, developed by Boyden. It's a very similar principle. And I would like to make this expansion microscopy be included as well in clearing methods because it's embedding the tissue in a hydrogel, but instead of expanding a little bit, you can expand loads more, and you do have very little protein loss with this approach. You can expand between 4 times to 25 times your structure. So you can artificially increase your resolution to nearly, in some cases, electron microscopy. So this approach can be very helpful for some people. If you're especially dealing with the tissue slices, organoids, etc., I would recommend this type of approach. Not for a whole brain, but for some smaller structures. It can be very valuable. So, as I mentioned, how do we decide which is the optimal approach, which is the optimal clearing method for my research? So, very important is to think about size, fluorescence, etc. And there is no published protocol that will match your method exactly. And I would recommend everyone to see this as clearing modules. I mentioned a few times already, they seem different, but they're all based on similar principles, which I'm going to go in a minute. Labeling, removing the lipids, refractive index matching. And these are modular. It's a concept is relatively new in the field. You don't need to follow water-based alone or solvent-based alone. You can mix match some of the steps. And I do that routinely in my facility. I start, for example, with a solvent-based approach, and I end with a final bit of the slightly different protocol. You can mix match to see what is more suitable for your samples. I am putting this summary out there, even in different blue color, just to mention hydrophobic or the organic solvents, basically RatDISCO, any of the DISCO methods. If your sample needs antibody labeling, go for it. It's great. The protocol is fast. Tissue might shrink. That could be an advantage or disadvantage, depends on the original size of your sample. But your samples will last forever. You can re-image them several times, particularly if you have a very delicate transgenic animal in a large cohort of behavioral animals that trying to repeat the behavioral cohort will be very difficult. You try to reduce the number of animals you're using, reducing the cost. You don't have to rerun the experiment. You can just go back and re-image parts of the brain that you need for a different research question. So I would say these methods, the solvent-based, are suitable for large and medium-sized samples. For the hydrogel embedding methods, I would recommend clarity in general. Um, I would say these methods with hydrogels and polymers are not suitable for immunolabeling. So if you have a transgenic, yes. If you do not have a transgenic, it's probably not the right approach. Tissue is very fragile, it will expand and it will collapse. So once you do the image, 6 months down the line, your tissue will be completely unsuitable for re-imaging. So it's only good for small samples and samples you can just produce often. And finally, the hydrophilic tissue clearing methods, which are water-based solutions. Those are non-toxic if you don't have in the lab equipment to deal with solvents. Water-based solutions, this is a good method. This cubic is a very well used and tested all over the place. Out of those 100 or so methods I mentioned, I am showing here some of the most reliable and the tissues clearing methods that most people use. Basically, they are reproducible. So the water-based or hydrophilic tissue clearing is compatible with immunolabeling. It can be slow. It might take a while, the slow processing times for the protocol, and it has a bit of limited performance. So some antibodies may not work in this approach. However, it's not toxic. And again, I would say this is suitable for large and medium size samples. And finally, I'm going to talk about the tissue processing steps so you can take modularity, the concepts I'm talking about, and a very brief view of the RatDISCO clearing. So all the tissue clearing methods, what they want is transparency. And by removing lipids and protein or pigments inside and matching the refractive index medium is how you achieve transparency. And as a hardcore anatomist and a histologist I've been trained like that for a lifetime. Please fix your tissue properly. People sometimes think it's the clearing method or antibody labeling. If you do not fix your tissue properly, nothing will work. So as a good anatomist, go back to the beginning. Never use all fixative. Always use freshly made paraformaldehyde. If you can do it at home, is the best. If not, if you have no choice, use formalin. But formalin is just a saturated solution of formaldehyde. And I don't know if you can see in my slices in here. Some formalin solutions can contain methanol and other type of alcohols to preserve the formalin, and that may inhibit immunolabeling. So if you can, do not use formalin. Perfuse fix or fix drop in freshly made fixative. Sorry, I always bring this slide because fixative is essential. After you fix your sample properly, as best as you can, then there are treatments. For example, they will help your clearing. One is removing the pigments with decolorization strategies like peroxide. There are quite a lot of solutions out there to bleach your tissue. Rehydration, most of the dehydration happens with solvents in one way, or for water-based, you rehydrate with urea or sucrose. Sometimes you can decalcify the tissue if you want a clear bone. And finally, this is the part that matters the most. Lipid removal. You can remove your lipids with soap, with solvents, with many electrical currents, with many ways. But removing the lipids is the most important part. Of course, labeling is optional because you might already have a transgenic mouse and you don't need that. And the final part, refractive index matching, which I am going to show right now. Sorry, I'm skipping one. Refractive index matching basically is you need to match the speed of the light passing through true mediums to make it transparent. And it sounds I'm trying to make it zero technical. So Imagine you have a beaker of glass with a solution. Both glass and that solution will have different refractive index. That's why you see this is transparent and this is yellow. So if you want to see this glass, if you want to matching the refractive index between these two glasses is embedding this glass on another glass. If you match with the same solution, this yellow solution in here, you can see the second glass disappears. It's not like the glass is not there. You can see through because the light has been matched, the speed of the light has been matched between those two mediums, between these two glasses and that liquid. And that's how you achieve refractive index matching. And the trick is just basically moving to a solution that it will be compatible either with your microscope or your final step of the clearing tissue. And gonna very briefly talk about RatDISCO, if I still have a couple of minutes out there. It's a method we just recently, it's in bioRxiv if anybody wants to take a look. We also always happy to share the protocol, bench protocol as it is. And because rat tissue is particularly huge compared to other mouse, zebrafish, even human tissue can be just obtained in small blood blocks, we decided to go old-fashioned anatomy histology again and go back to the 70s antigen retrieval protocols in which they use soap to denature proteins, high or alkaline pH to denature proteins, and heat incubation. If you denature the proteins, you open up the tissue a little bit, and the antibodies can penetrate much deeper and recognize sites that had been hidden in your antibody. After that optimization, that steps takes 1 hour. We follow then the traditional DISCO methods with some modifications in which again we rehydrate the tissue. We carry with alcohol with methanols. We immunolabel the tissue. By this point, the tissue is much more open thanks to antigen retrieval steps. So antibodies penetrate much deeper and they can see better the antigenic regions that you're looking for with your antibody. And then we remove the lipids with much stronger solvents and put the tissue to the light sheet microscope. And I want to show you a couple of examples of it. This is C4 immunolabeling in the amygdala of a rat, an animal that has been exposed to food shock. Or we have obviously the control animals that they did not. So you can see how we can fly through the entire amygdala of this rat. And then our objective at the time was to quantify the number of C4 positive neurons in animals, in the amygdala of animals. They had different type of models of autism versus control litter mates. I'm not going to show any data here. Everything's in the paper if anybody is interested. I am very happy to share the data and we have presented in conferences, but just to show you how these images look like. And after that, we can get this information in a much more, not visual, but actually a way to quantify this information is putting those three-dimensional amygdala of the rat and quantify it semi-automatically, automatically with software and quantify number of neurons in different parts, basolateral amygdala, central amygdala, etc. Obviously, that is a research question for autism spectrum disorders, but I'm going to show you with light sheet microscopy of other samples that we have in the lab. For example, in this case, I'm showing MECP2, which is a marker, well, it's the protein that is not functional in cases of Rett syndrome and other model autism spectrum disorders. And in the right side, you have tyrosine hydroxylase, labeling for all the dopaminergic, adrenergic, and noradrenergic neurons. And of course, in Parkinson's disease, these neurons will be lost. We have, I think it's the same one, this one. Using our RatDISCO clearing, we can also identify not just neuronal populations, but we can identify glia. In this case, I'm showing isob4, which is a lectin. And this is a case of a binding protein. A lectin is not an antibody, it's a lectin, it's a chemical bound that happens in there. We can also identify other populations that are not just neurons. We can also have a microglia in this case. I'm showing an example. We can also clear not just rat brains, but we can have rats' hearts, which are huge, they're much bigger than the rats. And we can also label very, very small things. In this case, is the pituitary gland of a mouse, which is the size of a grain of rice, and some organoids. In this case, these two organoids are actually so they are spheroids in purple or magenta, and in blue you have glioblastoma cells. And depends of how the tumor, if the tumor is disrupted, you can see how it completely invades the spheroid. And if the tumor cells are just located, encapsulated in one single structure without being disrupted, basically the spread of this glioblastoma is not so extensive. And this is for a model of a group that is working of glioblastoma is one of the most dangerous type of cancers in the brain, and that they are trying to study and just metastasis with this. So in summary, tissue clearing and light shield microscopy is helpful for imaging large-scale structures such as whole brains, organs, whole mouse, etc. As I say, our RatDISCO as well as every single tissue clearing protocol can help you identify neuronal bodies, blood vessels, glia, etc. And my advice is if you look at the size of your sample, look at if you need lipid staining or immunolabeling, and which especially once you clear your samples, which microscopes do you have in-house? Because you can be very successful in your clearing, but if your institution doesn't have the adequate light shield microscope or the adequate equipment, it's going to be very difficult to actually visualize the structures that you want. There's no point to have a whole mouse brain or a whole rat's brain. If you're going to look at this bit in the confocal, you need to go to a microscope that actually can look what you need. And that would be me. This is, thank you very much for your attention. I lead the light sheet microscopy facility at the University of Edinburgh. These are my email and QR code if you want to look at our facility. And the team is, the two women team in the lab is me and my technician, Kirsty Craigie, who does amazing clearing, and she's such an expert in all this clearing, imaging, and 3D analysis. And thank you very much for your attention.
Perfect. Thank you so much, Christina. That was a super interesting talk, and also super helpful and useful for anyone who wants to start doing tissue clearing. So thank you so much.
Thank you.
We have time for some questions. So if you haven't already, please pop your questions in the Q&A box. But we already have quite a few, so we'll get started. So our first question is somebody wanting to know if you have experience with fish methods along with clearing methods.
Fish methods?
Yeah, so fluorescent in situ
Fluorescence.
hybridization. Yeah, yeah.
Yes, actually, I did not show images in here, but RatDISCO is compatible with, we use RNA-scope just to make it fast, but it is compatible with normal immunofluorescence and RNA, basically RNA is immunohistochemistry. RNA-scope is the one we were using. But I know definitely Clarity that they is published. I haven't done it myself, but it's also compatible with in situs. And for us, it's a huge advantage if a protein doesn't exist, but you can label the RNA. It's possible. It's compatible with our method.
Yeah, perfect. Thank you. Our next question is how do you store the tissues that are cleared with different methods and labeled with antibody? And also, how long can those tissues be stored for?
For example, any of the DISCO methods, iDISCO, UDISCO, RatDISCO, they at the end they store in something. You can store them in solvents, which are toxic, or we can store them in ethyl cyanamide, which is not toxic. Actually, it's used for food additive, and it has the same refractive index of the 1.56 of the final solvents. They will last in room temperature. So far, I can tell 8 years because it's 8 years the samples we've been imaging. It will last forever, literally forever.
Wow.
Water-based tissue doesn't, if you keep it in the solutions of they will disintegrate unless you bring them back to PBS. And basically, they go back again to be opaque, but they can stay much longer in PBAs with ASI. And if you want to reimage them, you will have to re-clear them. But I would, for long-term storage, ethyl cyanamide at room temperature and forever for solvent-based clearing.
Wow. That's great. I wish some of my samples had have lasted for 8 years.
Yes.
Awesome. The next question is are a bit more general. So, are there any common mistakes you see labs make when they first start using tissue clearing that significantly affect image quality or reproducibility?
Mm, I would say obviously fixation. Start with a good tissue. If the tissue has blood or is poorly fixed, it will not clear well, and the antibodies will not work. For antibodies, for clearing, mistakes a lot happen that people will get an antibody that just bought recently. I will go to the literature, especially papers they had clearing methods normally publish the antibodies that are compatible with their clearing methods and which ones are not. For example, for my method, most of the antibodies need to be compatible with the methanol dehydrations. So and in our paper, we publish a large list of which ones they work and which ones they do not work. And most people will do that. So instead of choosing a paper for just your research, choose a paper, look a paper with the clearing method you want and look at the list they publish. And I would say finally the timings. I think sometimes people don't clear long enough. So the solvents, the protocols are 1 hour or two per solution. But if the tissue is bigger, just wait or exchange the solution and add more. I would say don't be afraid to modify the protocol to extend the incubation times or reduce, depends, to allow enough clearing. And I would say temperature. If it's cold, it won't clear very well. Most of the solvents and even every method is at 37 degrees or above. So room temperature 37 degrees or hot is good. Fridge 4 degrees is really not good for clearing.
Yeah, thank you. That was a lot of super helpful advice there from one question. Uh, awesome. So the next question is, when using electricity to help quicken processing time of hydrogel-based tissue clearing, how do you overcome inconsistent or uneven clearing, especially in larger samples?
I don't think there's a good solution for that. And I've never been a huge advocate of that method myself. I would say if you can, don't do the electricity. If you can, if you have the time, wait and use the passive one. If not, I would say change your clearing method. Move to water base if you want to, or solvent base.
Yeah, that makes sense. The next one is, hi, thank you for the great talk. Do you have any experience with clearing human tissues?
Yes, actually, we do actually have clear, because in the facility we receive samples of so many other groups. We have received, sounds going to be interesting, human leg samples from amputated tissue from diabetic patients. And it's quite a precious tissue. So we have no chance for error. So we normally recommend for human tissue, I would recommend solvent based because it would last forever, because it's a precious sample that you can image and re-image and re-image. And I'm not sure if I mentioned, but solvent, you image to the immuno image. You can rehydrate, re-immunolabel against something else, re-clear and go back to image. So you can do multiplexing. You can use 4 or 5 channels and then go back and multiplex and multiplex. Obviously, by the third round of labeling, you might not get the great signal. But because the are precious samples, I would say solvent based. Our protocol, we have that and we have 1 or 2 samples of human brain. But yes, we do skin, muscle, and brain.
Nice.
And happy to
Um,
help anybody who wants, they can email me if they want and we can share our protocols. No
oh,
problem.
fantastic. Great, thank you. The next question is, do you think shelf-stable PFA solutions are comparable to freshly made powder-based ones?
Pro, can you say it again, please?
Sorry, so do you think shelf-stable PFA solutions are comparable to freshly made from powder-based?
I would say no. The only ones I would say comparable as the electron microscopy grade formaldehyde,
Yeah,
but those samples are small and very expensive. And it's okay if you perfuse a mouse, but it will be very expensive to perfuse a rat because you need a much larger volume. So I would say make it for fresh. If it's
yeah,
cheaper and is yeah.
yeah. Don't be lazy, guys. Just
It's
make
also
up.
much in an emergency. I get it, if especially for a mouse. EM
Yeah,
grade is fine. It
yeah,
will be cost effective, not wise if you try to produce a larger sample.
yeah. That is very true. So somebody also asked, what would be a good technique for clearing brain organoids, which we would also like to immunolabel?
I would say go for the water base or the solvent base. We have used normally that there is modify. I can email this the reference Klimberg et al. Paper with ethyl cyanamide. It's a really simple protocol. Ethanol dehydrations match at a pH 9. Immunolabeling, ethyl cyanamide clearing, because the samples are so small, the delipidation is achieved with simple ethanols. In 2 days is achieved. I would recommend that, or yeah.
Nice. And then I think this will be our final question, unless anyone wants to add any more to the Q&A box. So this is a question that's more related to quite specific downstream application.
Mhm.
So if tissues or cells are fixed with PFA, they were wondering if you have experience with that affecting downstream next generation sequence library construction, because they're thinking about maybe cross-linking might impact the DNA quality or amplification.
Uh, I don't know. I mean, that I don't know. The cross-linking.
Yeah.
One way we do, for example, not for DNA, but some antibodies don't like overfixation, especially CFOS. We published that in our paper. 3 hours post fixation, any longer, and the CFOS immunolabeling can be unreliable. Overnight post fixation is not a problem for parvalbumin, GFAP, GFP, not an issue, but over cross linking, it can affect. So I would say reduce the fixation times to test what would work best. Perhaps only 1 hour, perhaps 3 hours, but not overnight, perhaps would be ideal.
Yeah, yeah, that makes sense. So we actually got another question whilst you were answering that last one. For labs without access to light sheet microscopy, which tissue clearing approaches still provide meaningful benefits when using confocal or other systems?
Well, confocal is not suitable for solvent clearing generally because if you want the best image, you don't want to pass through air, which is a refractive index of 1, then through glass, which is refractive index of that I don't know, then to the water solution, which is refractive index from 1.33 water to up to 1.46, which is the water-based approaches. So you're passing through all these different indexes, and obviously you will get distorted images. So you want an objective that is immersed in the liquid. So the objectives in confocals can only be immersed in water base. There is a very good 20x objectives, water base. They don't necessarily need to be clearing objectives. They could be the normal 20x water dipping objectives. But then for that, ideally, you need to use the water base clearings unless you protect it. I think there is you can protect it with glass if you go for solvents, but if you have a light sheet, solvents. If you don't have a light sheet, water based and a confocal. But the huge advantage of the solvent-based clearing is that you can ship your samples across the world to be imaged. In our facility, we have received samples from Ireland. They just put them in the post and they send it to us, and we image them because they are solid, and then that's fine.
Wow, nice. Awesome. I think that was the last of our questions. So if anyone doesn't have any more, I'll give a couple more seconds just in case anyone has a last minute question that they're dying to put in the box.
Yes, and in the facility, as I say, we're always open. Send us an email. Before we publish our paper, we share the protocol with absolutely everyone. Please send us an email and we share our adaptations and we can give you tips. It's not an issue. We like to share and make it more reproducible.
Oh, fantastic. No, that's brilliant. Spirit of collaboration. Yay.
Yeah.
Awesome. Well, I think, yeah, that's the last of our questions. And again, thank you so much, Christina, for such a interesting and useful talk, and also for answering our barrage of questions.
Thank you
Everyone was
so much.
clearly very interested. So yeah, thank you so much, and thank you everyone for attending. And yeah, that will be the end of our first Gene Delivery Seminar series for 2026. So thank you so much, everyone, and
Thank you.
goodbye.